Okay, chapter 65, organization of the gastrointestinal system. So what are the alimentary tract, it provides the body with a continual supply of water, electrolytes, vitamins and nutrients. So this requires a few processes. So what are they? So your food needs to move through the alimentary tract, that is a motility function, and then the digestive juices, they need to be secreted so that they can digest your food, so that you can absorb the water and various electrolytes from the food. And then you need to circulate these absorbed nutrients through the blood to the organs. Okay, and then you need to control all these functions by local nervous and hormonal systems. Okay, so when we talk about the general principles of gastrointestinal motility, we need to talk about the layers. Okay, so what are the layers? As you can see here, from outside to inside, you have the serosa. Okay, and you have the mesentery, it encloses the mesentery. Then you have the muscularis. Okay, in the muscularis you have outer longitudinal muscles and inner circular muscles. Okay, between the outer longitudinal and inner circular muscles you have your myenteric or Auerbach's nerve plexus. And then after the circular muscles, inner to it you have the submucosal layer. Okay, and in the submucosal layer you have the submucosal nerve plexus or the Meissner's plexus. Then you have the mucosa. Okay, so the mucosal lining you can see here, the epithelial lining of the mucosa, the mucosal muscle lining, muscularis mucosa, and then you can see the mucosa, and then you have certain glands in the epithelial lining of the mucosa. This is your lumen. Okay, and then you also have some submucosal glands such as Brunner's glands in the duodenum. Now we have to understand that sparse bundles of smooth muscle fibers, the mucosal bundle, they lie in the deeper layers of the mucosa. So what are the types of smooth muscles? We have multi-unit smooth muscles and single unit smooth muscles. So multi-units, that means there are many units of smooth muscles. Each unit has separate innervation by a single nerve ending, and each fiber operates independently of the others. Okay, it is composed of discrete smooth muscle fibers. So you can see the diagram, all of them are discrete smooth muscle fibers. Okay, whereas unitary smooth muscles, they are also called syncytial smooth muscles. They are a mass of hundreds to thousands of smooth muscle fibers. They contract together as a single unit. So you can see here. Okay, and then so the gastrointestinal smooth muscle, it functions as a syncytium. So in the longitudinal muscle layer, the bundles extend longitudinally down the gastrointestinal tract, and in the circular muscle layer, they extend round the gut. Okay, within each bundle, all these cells, they are connected electrically by gap junctions, which act as low resistance bridges. Okay, they allow the movement of ions from one muscle to the next, allowing all of them to contract simultaneously at the same time. Each muscle layer, it represents a branching lattice work of smooth muscles, and it functions as a syncytium. So when an action potential is elicited anywhere within the muscle mass, it generally travels in all directions of the muscle. Okay, so the distance it travels depends on the excitability of the muscle. So excitation, because of a few connections between longitudinal and circular muscles, excitation of one of these layers often excites the other as well. So if you want to describe the electrical activity of the gastrointestinal muscle, you can do that in two waves. One is you can describe the slow waves and the spikes. Okay, so as you can see in this graph here, this represents the slow waves. So your slow waves, they are not action potentials, they are just slow undulating changes in the resting membrane potential. Okay, what are the values? They range from about 3 per minute in the stomach. Remember this value, 3 per minute in the stomach. And then in the duodenum, they are about 12 per minute, 12 waves per minute. So you can remember it like this. Duodenum comes from dodecadactylos, which means when it was first discovered, it was about 12 fingers in length. So dodeca, 12, okay, 2 plus 10. So it is about 12 waves per minute. And these slow waves, they are caused by complex interactions in the smooth muscle pacemaker cells in the GIT, that is the interstitial cells of Cajal. Okay, they undergo cyclic changes in the RMP because of pacemaker current. Okay, so they have unique channels that open and close, and due to inward flux of pacemaker current, these changes in the RMP are produced. The slow waves themselves do not cause muscle contraction except perhaps in the stomach, because they don't cause calcium to directly enter into the smooth muscle fiber. Okay, so this is about the slow waves. And when we talk about the spike potentials, as you can see, one important point is higher the slow wave potential rises, the greater the frequency of spike potential becomes, ranging from 1 to 10 spikes per second. So the slow wave potential, as it rises, the frequency of the spikes also increases. Okay. So the spike potentials, they are true action potentials. They are caused when RMP rises above -40. So what is the difference? In nerve fibers, action potentials are due to sodium influx, okay, the depolarization process. This is via the sodium channels. However, in GIT smooth muscle fibers, remember that the wave you see here correlates with the contraction of the muscle. So that means it should have something to do with calcium, because the muscle is contracting along with the wave, okay? So the calcium influx is due to calcium sodium channels, which are longer to open and which take longer to open and close. So hence the duration of the action potential is increased. It is maintained at an increased rate. And this calcium influx helps in contraction of the muscle fibers in relation to the wave. So how does this occur? Because they are smooth muscles, the calcium ions after the influx, after entering into the smooth muscle cell, they act via calmodulin control mechanisms. They then activate the myosin filaments, which develops attractive forces between the actin and myosin filaments, and finally, as a result of that, your muscle contracts. Okay. So let us talk about changes in the voltage of the RMP. Okay. So the RMP under normal conditions, it averages about -56 millivolts. Okay. But multiple factors can change this level. So when the potential becomes less negative, when it rises, okay, it is called depolarization of the membrane. Muscle fibers are more excitable. When it becomes more negative, when the RMP is negative, it is called hyperpolarization. Fibers become less excitable. This is the basic thing. So factors that depolarize the membrane include stretching of the muscle, stimulation by acetylcholine from the endings of parasympathetic nerves, and stimulation by gastrointestinal hormones. Always remember, parasympathetic stimulation depolarizes your GIT. It increases your motility. Acetylcholine, it increases the rate of gastric juice secretion and all that. Okay, parasympathetic is positive in case of GIT. Okay, causes positive effects. So as you can see, stimulation by stretch or acetylcholine or parasympathetics, it increases your slow wave potential, and also it causes increase in the frequency of spikes. And however, if you use sympathetic stimulation or release norepinephrine at the nerve endings, it causes depolarization. Okay, sorry, hyperpolarization. Okay, so that is about it. And so factors that hyperpolarize the membrane, make it less excitable, are effect of norepinephrine or stimulation of sympathetic endings. And we know this entry. Okay. And then what is neural control of gastrointestinal function? We have an enteric nervous system in the gastrointestinal tract. Okay, it is a nervous system, all of the own of the enteric gastrointestinal tract. It is comprised of two plexuses. So one is the myenteric or the Auerbach's plexus that lies between the longitudinal and circular muscle fibers. Okay, you can see here. The other is the submucosal plexus. Okay, you can see that. Submucosal plexus in the submucosa. It is called the Messner's nerve plexus. Okay. So the nervous connections they connect both these nerve plexuses. Okay. So as you can see from this very high yield diagram, your sympathetic post-ganglionic nerves, the red fibers, they innervate all these structures, both the myenteric plexus and the Messner's plexus. Okay, and the parasympathetic also, they innervate. The parasympathetic fibers that innervate them are preganglionic, because the postganglionic fibers, they are a part of the enteric nervous plexuses. Okay, so like this they innervate. And you also have sensory fibers that arise from the epithelium of the gastrointestinal tract, and they further give connections to both the plexuses as well as the prevertebral ganglia, spinal cord, and the brainstem. Okay. So this is the neural control of the gut wall. It is a very high yield diagram. Okay. So although remember this, that the enteric nervous system, it can function independently out of these extrinsic nerves. Okay. But the stimulation of sympathetic and parasympathetic nervous systems, it increases or decreases the gastrointestinal functions. Okay. Otherwise, the enteric nervous system is enough to sustain the functions. And then there are sensory nerve endings that originate in the GIT epithelium. They send afferents to both plexuses as well as to the prevertebral ganglia, spinal cord, and the vagus nerves in the brainstem. Okay, these sensory nerves are useful for eliciting local reflexes within the gut wall itself and other reflexes that are related to the gut from either the prevertebral ganglia or the basal regions of the brain. So when we talk about types of neurotransmitters, again we have several types of neurotransmitters, but remember again, acetylcholine excites gastrointestinal activity and norepinephrine and epinephrine inhibit gastrointestinal activity. And then when we talk about the differences between the myenteric and the submucosal plexus, the myenteric plexus, myo, the name indicates it is concerned mainly with controlling muscle activity. Okay, so how does it do that? It increases the tone of the gut wall, increased intensity and increased rate of the rhythm of contraction, and also increased velocity of conduction of excitatory waves. And it is composed of both excitatory as well as inhibitory neurons. So this is important because you have something called VIP, vasoactive intestinal peptide. So it is a parasympathetic, it is done by it has parasympathetic effects, but in the name you have inhibitory. So why is that? It actually decreases the motility at the valves. Okay, so your valves are opened. So what happens is your feces, they can pass open nicely through the valves. So motility, it is not decreasing. Okay, so that is the thing. And in the submucosal plexus, you mainly have excitatory neurons, and they are concerned with local absorption, local secretion, local contraction of the smooth muscles. And then the parasympathetic stimulation increases the activity of the enteric nervous system. It is a craniosacral outflow. So the cranial parasympathetic nerve fibers are almost entirely in the vagus nerves. Okay, they extensively innervate the esophagus, the stomach, the pancreas, etc. The sacral parasympathetics, S2, S3, S4, remember this, they originate from these segments of the spinal cord. They pass through the pelvic splanchnic nerves to the distal half of the large intestine and all the way to the anus. So they extensively innervate the sigmoidal, rectal and anal regions because defecation has to take place there. Okay. And the postganglionic neurons are located in the plexuses. So stimulation of these parasympathetic nerves increases the activity of the entire enteric nervous system. Then what about sympathetic stimulation? It inhibits gastrointestinal tract activity. They originate between segments T5 and L2. Most of the preganglionic fibers, they innervate the gut after leaving the cord. They actually enter the sympathetic chains that lie lateral to the spinal cord. Okay, and most of them, they pass to the outlying ganglia such as to the celiac ganglion and various mesenteric ganglia. The sympathetics innervate essentially all of the GIT, okay, rather than being most extensive here, most extensive there, something like that, as in the case of parasympathetics. They release mainly norepinephrine and also epinephrine. They inhibit the activity of the GIT, okay. So how does it inhibit the activity? It has a direct inhibitory effect, that is norepinephrine. It has direct effect on the intestinal tract smooth muscle. It inhibits it, and also it has an effect on the neurons of the enteric nervous system, on the smooth muscles and also the neurons in the GIT. So what about the afferents? So we talked about afferent sensory fibers that innervate the gut. We need to understand that 80% of the vagal nerve fibers are afferent. They are not efferent, okay? Because gathering information from a body as huge as ours is important. Only when you know the complete details, you can act properly, okay? So these sensory nerves, they can be stimulated by excessive distension of the gut or irritation of the mucosa or presence of chemical substances in the gut, okay? So they have their cell bodies in the enteric nervous system and some also in the dorsal root ganglia of the spinal cord.